name is Dr. Jonathan Stein, and I'm the CSO and Lab Director for SpectraCell Laboratories. I'm delighted to let you know we are now offering COVID-19 antibody testing, and we put together this small presentation to let you know some of the particulars about it. First, a little bit of background. SpectraCell Laboratories has been around for a good long time, heading towards 30 years. We were the first laboratory to offer functional micronutrient testing, which is a test that just tells how your immune system can be optimized through nutrition. We also added novel cardiovascular disease risk and anti-aging markers, as well as standard hormone and nutrition tests. My background is in, basically, I've been a geneticist my whole career. My undergraduate's in biochemistry and molecular biology from UC Santa Cruz. I also went to the University of Texas and got to study virology, molecular and population genetics. My postdoctoral training was in cancer research, did a little bit more virology in there. I went on to Adenogene to do molecular forensic testing and paternity testing, and then came on to SpectraCell as the science director, and then lab director, and then CSO, where we do clinical diagnostics, essentially specializing in nutrition, endocrinology, and chemistry. I need to say that we are scientists here and not clinicians, so we cannot provide clinical treatment advice. We do the diagnostics. We are a CLIA accredited diagnostic laboratory. So whenever your doctor collects a sample and sends it off for testing, we are the testing part. When the results come back to your doctor and they make clinical recommendations based on that, that's not something we can do. So we do the testing, we're accredited to do that, but we cannot provide treatment options. Very briefly, you'll hear bandied about a lot of nomenclature around this pandemic. Very briefly, the coronavirus is the family of viruses that this particular disease is coming out of. It's named for its crown-like shape. SARS-CoV-2 is the name we're going to be giving for the virus. Severe acute respiratory syndrome coronavirus 2. And then you'll also hear COVID-19, the disease that the virus causes. First appearing at the end of last year in China, first appearing in Washington in early 2020. The sequence analysis, independent sequence analysis of the virus strongly suggests a natural origin, and that was reported in one of the top scientific journals in the world, Nature, in April of this year. The infection rate, it has varied in different populations. Obviously, behaviors have been, such as sheltering in place, have been performed to hopefully reduce that infection rate. But the initial studies thought that it had a basic reproduction number of about 2.2 to 2.7. Newer studies suggest that it's much, much higher than that. There have been suggestions of multiple strains. Obviously, viruses create different copies of themselves all the time, and they may have different infection rates. That's not surprising. It's also true that many different factors can affect someone's infection rate. Cultural behavior as simple as shaking hands versus bowing versus hugging and kissing and greeting can increase or decrease the infection rate of the virus. Obviously, mass transit, air travel versus individuals traveling in cars will impact the number of people an infected person could potentially come into contact with. And population density is going to matter as well. The disease is primarily spread through close contact, small droplets when people cough, sneeze, or talk. It can also be spread through fomite transmission, so touching a contacted surface. Also is present in contaminated water, and in circumstances in countries where public sanitation is a big issue, this may also be a very large source of potential infection. The fatality rate has pretty broad error bars, since the number of people infected is sort of the denominator of that calculation. The more people who are infected, then the smaller the fatality rate will be for any individual person. what is known, so it's got fairly large error bars by an order of magnitude. But we do know from the various public health studies that have come out of Italy and Israel and China and the US now that the fatality rate increases with age. Going into a little bit more detail, here is some data from a New York study as of April of this year. You can go onto this website link and see some of the demographics of it. It's worth saying many of these articles haven't yet been peer-reviewed. The disease has only been around for a few months, and the peer review process for publication usually takes about 6 months. I know people are rushing out papers, but you know, everyone is hurrying as quickly as possible, but it's worth bearing in mind. So this was a study on, I believe, about 7,000 people out of New York. If you look at the age bracket, so it shows the age bracket, the numbers of deaths in each cohort, the percentage of deaths out of the total number of deaths, the percentages that had known underlying illnesses, and then the share of deaths, including individuals who either they couldn't get a health history on or had no conditions. And what really jumps out here is for individuals below the age of 45 for the New York study, very, very few people without pre-existing conditions succumb to the disease. This is supported in, I believe, the Israeli study. I think 96–98% of the deaths were over 65. In the Italian public health data, I believe the mean age of fatalities was 79.5 with pre-existing conditions. I know that in the Israeli data, nobody under 20 without a pre-existing condition had died of the disease. So that's good news for those of us who are parents with small children. This disease primarily seems to be impacting individuals over 65 with pre-existing conditions. And you can see that again in the share of deaths here by age. About twice as many males as females, just going to that far right part of the slide. 62% or 61.8% of males constitute the majority of fatalities, women about 38%. So that's what the demographics of this illness look like. Many of the people hospitalized suffered from obesity, so people hospitalized for the illness independent of the disease. I've seen estimates of about 50% of the hospitalized cases being people with obesity in one study, or at least one clinical observation. Underlying illnesses included diabetes, lung disease, cancer, immunodeficiency, heart disease, hypertension, asthma, kidney disease, and gastrointestinal and liver disease. So those individuals who are over 65 with those conditions could be considered the highest, the group at highest risk for mortality from this disease. Here are just some of the symptoms and the rough percentage. This was pulled off the CDC's webpage. Again, this information is coming in fast, but this is the information from April of 2020, so it's pretty recent. That said, it's a very fluid situation. Summarizing it, I'd say that it looks like it's a head cold unless it gets into the lungs, and then it looks a lot like progressive pneumonia. So we are performing antibody testing. There's also molecular testing, and I just wanted to highlight some of the differences between those two types of tests. So the nucleic acid or molecular testing is a test that looks for viral RNA in epithelial tissue. So that's the one where they're using a nasopharyngeal swab, going up the nose very, very deeply, trying to collect a good epithelial tissue swab, and then extracting the RNA from that sample, converting it to cDNA, and looking for the presence of viral RNA in that sample. That's a very high complexity test. It's hard to get good samples from a nasopharyngeal swab. It's hard to handle RNA. So there have been reports of some false negatives coming out of either a poor sample collection or laboratory errors caused by this very high complexity assay that people have worked heroically to get into production. I'm not criticizing anybody who got this up and running in the laboratory, just that it's a very, very challenging assay. By way of contrast, we're doing antibody testing, which essentially looks for tries to identify antibodies to the virus. So that's the body's response to the infection. It looks for human antibodies to the virus, and looking for long-term antibodies. The virus will have already passed, of course. And I'll show you a slide showing some of the time course on this. It's a blood test. It's very, very straightforward. A finger stick or a small blood draw. The assay itself is fairly straightforward. It's comparable to a pregnancy test. Blood is put on a strip that has a colorimetric response to the antibody. It's either present. The antibodies are either present or they aren't. So the test itself is categorized as medium-high complexity test. We could perform both of these, being a high complexity CLIA-approved laboratory, but we thought that antibodies would be much faster to get to the consumers. So this is a slide from Abbott Laboratories, and it's just an estimate, and it's the one they're using to describe their antibody testing. So there's this period from about 5 to 7 days in which a person might be infected and have a very low viral titer, and so they may not have an immune response to that. We're going to be testing for both IgM and IgG. So if you see the sort of x-axis of the graph, it's just the days since infection, and it's just an estimate. But it's thought that for the first 5 to 7 days, a person might have detectable virus in their system, but they wouldn't have a detectable immune response to it yet, just because the viral load hasn't become high enough that it's provoked an immune response in the host of the virus. So it takes a little bit for the IgM to become detectable, and then that IgM does its thing, so it's an initial immune response. And then a little bit later, IgM will start recruiting and inducing an IgG response. So that's the secondary long-term antibody that is thought to provide some resistance to infection or reinfection, and that's great. And then IgM will slowly disappear, and then IgG will hang around and again provide that hopeful immunity, but at least a strong level of resistance to reinfection. Lots of stuff humming around lots of stories moving around in the media. There have been reports in South Korea of reinfection that the Korean CDC came out and said that those were false positives, that the patients had fought off the virus, they had dead copies of, they had inactivated viral RNA in their system, and the molecular test, the nucleic acid test, identified the presence of those killed viruses. So again, it's the interpretation that can get kind of complicated. There may be cases where patients are immunocompromised or something like that, where there may be reinfection, but it looks like that's extremely rare. And based on the original South Korean reports, it looks like they were reporting false positives from the nucleic acid test. So again, it's the interpretation of these tests that are kind of complicated. Here we're going to be testing both the IgM and the IgG, and a person can be negative, so they're negative for both of those antibodies in their system, and that would mean that the person hasn't been infected with the virus, or they have not yet produced antibodies in response to the virus, which typically occurs very early in infection and prior to symptoms. The rough guesstimate on that is 5 to 7 days, but no one really knows for sure. So in all of these cases, the test itself should be used in conjunction with the symptoms that the patients are presenting. And again, your physician or your clinician would give you the best medical advice based on the results received. If someone is IgM positive and IgG negative, that suggests they're in the earliest stages of the infection, when their sort of initial immune responses happen, but that secondary response hasn't kicked in yet. If they're positive for both of them, so they're positive for IgM and positive for IgG, that suggests that the patient is in the active phase of infection, heading towards the tail end of that infection when the IgG has been recruited. And then IgM negative, IgG positive, that would be sort of the best outcome, and that would suggest that the patient is either on the tail end of their infection, so heading towards the end of it, their IgM has completed its cycle, or that they are no longer infected and now have some resistance or immunity to the virus. And so that I think, as the population accumulates of people who are IgG positive for COVID-2, for SARS-CoV-2, those folks will sort of present a sort of herd immunity that will reduce future infection. At least that's the hope. So in any case, all of these could be verified through a molecular test, right? There's lots of unanswered questions about this. It's thought to be an enormous pool of asymptomatic patients. The Icelandic study showed 50%. A blood draw in Lombard, Italy, of about 80 people showed that 70% were asymptomatic but were positive for the virus. There have been at least 2 reports this week in the U.S. of, I believe, a meatpacking plant and a prison where the population was 98% asymptomatic, the tested population. So those could be younger people, but also the more infectious the disease is, the lesser the sort of per capita fatality is. Not that any fatality isn't tragic, but I guess that question really hasn't been addressed yet. We don't really know, I guess, for high risk of severe cases for this question here, the data seems to be fairly concordant. The public health studies and reports that have been out looking at New York and Israeli and Italian and Chinese data, it really looks like the elderly and infirm are at highest risk. We discussed previously some of the pre-existing illnesses that look like they predispose the elderly for death due to the virus. So those are the folks who really, really, really need to be careful. The best time to get tested, again, those windows for maximum antibody response or when antibodies will become detectable, those really haven't been worked out yet. It's very early days. So that's something we'll be watching carefully, and we'll try to update as we get more information. The patient relapses, again, it looks like that is very, very rare based on the South Korea data from late last week, early this week. Reports of patient relapses may be artifacts of looking in serum for the presence of viral RNA, because that can be present with the virus dead. So again, early days on that. So I'm not 100% on that, and neither is anyone else. How long do IgG antibodies to SARS-CoV-2 last? Based on other coronaviruses, the guess is 1 or 2 seasons. Obviously, the virus will be mutating over time to try to evade the antibodies, and that's sort of an arms race. But I think a good model to use here is flus. In a good or bad flu season, last year's vaccines will still confer some resistance or less, but it's thought maybe 1 or 2 seasons, but it could be longer. It just sort of depends on rate of mutation and how that plays out. Again, the known coronaviruses typically last for 1 to 2 seasons. For some of the known SARS-related viridae, how long immunity lasts is simply unknown. the virus hasn't been long enough to be really determinative about that. So there are testing limitations. You know, people could get false positives or false negatives, and the virus could potentially mutate in the antibody target region, which could kind of confound the results as well. Just going through these really briefly, you could be an individual who has previously gotten, who has previously been infected from one of the other human coronaviruses, and it's possible that an antibody test could identify those antibodies. I will say we were fortunate enough to have a volunteer patient who previously was thought to be infected with SARS, who we knew was negative based on the molecular test. So they got the nasopharyngeal swab and did not have any detectable viral RNA in their system. And they came out negative on the initial antibody tests we're using. So that is promising, but that's just one individual. You know, it's entirely possible that there could be confounding factors about which we are not currently aware. This is just fast. You know, there could be test defects as well, but it seems very unlikely. We've tested our kits against 2 known positives and 1 known negative, and the results have looked good. You could be false negative if your antibodies are below the level of detection. And that could happen if someone is immunocompromised. There may be possible interfering compounds that could interfere with the binding of the antibody to its target. That's very hypothetical at this point, but it's certainly possible. You can imagine immunity waning over time, but that's not going to be an issue at this point in time, for sure. And again, it's entirely possible that the virus could mutate in the antibody target region. So that over time, these tests may need to be updated with newer, more specific antibodies. Probably not going to be an issue in the short term. The full epidemiology and clinical spectrum of infection caused by SARS-CoV-2 is not fully known. We talked a little bit about this, that somewhere between 50 and 95% of individuals tested may have no symptoms. So the results these tests provide may not correlate with signs or symptoms of disease. As with most diagnostic laboratories, these tests have been made under an emergency waiver, and the kit manufacturers have submitted these for FDA approval, but it'll be up to the FDA to determine how quickly they can get through the review and approval process. So they're not FDA approved at this time, but they have been made commercially available under an emergency FDA waiver. We are a CLIA accredited diagnostic laboratory, but we are not licensed to provide clinical treatment options. We're scientists, not doctors, basically, and so we perform the testing. But you'll need to take those test results to your clinician to discuss treatment options. And again, antibody results shouldn't be used as the sole basis for treatment. That'll be typically done in conjunction with the symptoms you're displaying, but that's up to you and your physician. Online on our webpage at www.spectracell.com, we've got an overview of the antibody test, some basic information, some technical information, and a glossary of these COVID-related terms. If you are interested in purchasing the test, you can visit us at www.spectracell.com/antibody test, and that'll get you that information or some contact information. So I hope this information is helpful. I hope everybody stays safe, and we hope to hear from you soon. Take care.